All of the first-generation vaccines teach the immune system to make antibodies against the coronavirus spike protein. The virus uses the spike proteins on its surface to attach to the ACE2 receptor on host cells. Neutralizing antibodies against the spike protein block attachment to the ACE2 receptor and prevent viral entry into host cells. Since SARS-CoV-1 and MERS coronavirus that emerged a decade later, scientists have known that the neutralizing antibodies against the coronavirus spike protein are protective. I suggest discussing vaccines to start with the family of vaccines that are called gene-based vaccines. They include messenger RNA, DNA, and viral vector vaccines. These vaccines carry the genetic instructions for the host cells to make the antigen, which more closely mimics a natural infection. The approach of gene-based vaccine isn't entirely unfamiliar. In live attenuated vaccines, like the measles, mumps, and rubella vaccines, weakened viruses incorporate their genetic instructions into host cells, causing the body to churn out viral copies that elicit antibody and T-cell responses. Now, among the family of gene-based vaccines, I will start with messenger RNA vaccines that are non-replicating. The current messenger RNA vaccines are non-replicating, which means the messenger RNA is unable to replicate itself. The two currently approved messenger RNA vaccines from Pfizer-BioNTech and Moderna consists of single-stranded RNA that contains the coding sequence for the full-length coronavirus spike protein, flanked by regulatory regions on either end. The single-stranded RNA is produced using a cell-free in vitro transcription from the corresponding DNA template. Modified nucleoside. The two vaccines contain a modified uridine triphosphate to make the messenger RNA less inflammatory. Indeed, a non-self nucleic acid in the cytosol activates the cell's innate immune system, which may cause a reduction of protein expression in case of repeat dosing. However, for vaccines, messenger RNA that engages the innate immune system may actually potentiate the response to the vaccine. Nevertheless, both the Pfizer-BioNTech and Moderna vaccines use a nucleoside-modified messenger RNA. Modified spike protein. Both messenger RNA vaccines encode for modified coronavirus spike protein that contains two proline substitutions to stabilize the spike protein in its pre-fusion conformation for better antigenicity. This is important for a safe and robust immune response. Encapsulation. The messenger RNA is encapsulated in a carrier lipid nanoparticle, LNP, that protects the messenger RNA long enough to reach the desired tissue, preventing it from degrading too quickly in the body. The lipid nanoparticle may even have an immune-stimulating adjuvant effect. In addition, the lipid nanoparticles help the messenger RNA cross the cell membrane and allow its uptake into endosomes. The messenger RNA is subsequently released into the cytosol, where it is translated into the spike protein. The spike protein then migrates to the cell surface. Immunological properties. The spike protein expressed on the host cell membrane is recognized by the immune system as a foreign antigen. This elicits the production of antibodies and the activation of CD4 positive helper T cells. In addition, CD8 positive cytotoxic T cells, killer cells, are recruited through the MHC, major histocompatibility complex class 1 pathway. As you might remember from your immunology course, MHC class I molecules bind antigenic peptides and present them to T cells. The MHC peptide complex is recognized by the T cell receptor and its CD8 co-receptor on cytotoxic T lymphocytes. This results in the activation of the cytotoxic T lymphocytes. Of note, only viral proteins produced by the host cells themselves are expressed through the MHC class I pathway. Thus, gene-based vaccines, such as RNA, DNA, and viral vector vaccines, possess the unique advantage of being able to activate CD8 positive cytotoxic T lymphocytes, which likely contributes to their high efficacy. Pharmacological properties. After intramuscular injections, cells at the injection site and the draining lymph nodes take up the lipid nanoparticle, effectively delivering the messenger RNA sequence into cells for translation into viral protein. The delivered messenger RNA does not enter the cellular nucleus or interact with the genome. It is expressed transiently, mainly by dendritic cells and subcapsular sinus macrophages. The vaccine messenger RNA is destroyed after a few days at most. Efficacy. The two-dose regimen of the two messenger RNA vaccines boosts the immune response and the generation of memory cells. For both vaccines, the interim analysis of their phase 3 data, respectively published in the New England Journal of Medicine, indicates an efficacy of about 95% against the circulating viral strains. Safety. In vaccine development, there are some general caveats to be aware of, in particular the possibility for an excessive or misguided immune response in vaccine recipients and the possibility of vaccine-enhanced disease upon encounter with the coronavirus. What we know is that in terms of tolerability, both messenger RNA vaccines cause local and systemic adverse events. Local adverse events include pain at the injection site, swelling, and redness. Systemic adverse events include fatigue, headache, nausea, arthralgia, myalgia, and the raised body temperature. I mean, not all of them together, but some of them may occur. Anaphylaxis remains rare, but appears to be more frequent with the messenger RNA vaccines than with the flu vaccine. Anaphylactic reactions may be attributable to one of the fat components contained in the lipid nanoparticle, that is, to polyethylene glycol. Polyethylene glycol is a molecule found in products such as cosmetics and laxatives. So far for the two non-replicating messenger RNA vaccines. There are, however, currently also self-replicating, self-amplifying messenger RNA vaccines in development. These contain messenger RNA that is able to replicate itself. Self-replicating messenger RNA vaccines are currently explored by BioNTech, Imperial College London, and other vaccine developers. With a conventional messenger RNA vaccine that is non-replicating, a lower dose means lower potency. Self-replicating vaccine candidates, however, include instructions for the RNA to copy itself. They contain a replicase gene, RDRP, RNA-dependent RNA polymerase, to do that. The self-replicating messenger RNA vaccines should allow to lower the dose. Also, these vaccines more closely mimic a natural infection, triggering a stronger, broader immune response. This in turn might even allow for single-dose inoculation regimens. Now, we talked about messenger RNA vaccines replicating non-replicating. What about DNA vaccines? DNA is a very stable molecule due to the double helix, and this stability presents important advantages for vaccine storage and transportation. DNA vaccines must enter the host cell nucleus. From there, messenger RNA is created, which travels out of the nucleus into the cytoplasm, where protein is formed from it. Thus, DNA vaccine technology faces two important challenges. One, the vaccine must cross the cell membrane, and two, the DNA must be delivered into the cell nucleus. The delivery of the vaccine DNA into the cell is achieved with an electroporation device, applying electric pulses to the cell membrane to make it become more permeable. For delivery into the cell nucleus, the vaccine DNA might include genetic information encoding for proteins that facilitate entry into the cell nucleus. Otherwise, the process may be inefficient. A potential risk associated with DNA vaccines is that vaccine DNA might be integrated into the genome of the cell. Another type of gene-based vaccines are the viral vector vaccines, and those currently in development are non-replicating viral vector vaccines, which means that they contain vector viruses that are unable to produce new viral particles. There is the Astrazeneca vaccine used as a two-dose regimen, the Johnson & Johnson vaccine used as a single shot, with studies exploring a two-dose regimen underway, and the Russian Sputnik V vaccine. All these vaccines use a common cold virus of the family of adenoviruses to transport the genetic instructions to build the coronavirus protein into host cells. Adenovirus are non-enveloped viruses that contain a double-stranded DNA genome. The vaccine's adenovirus DNA has been modified, both to be replication deficient and to contain the genetic instructions for the synthesis of a full-length coronavirus spike protein. These genetic instructions are inserted in the form of double-stranded DNA. The double-stranded DNA fragment encoding for the spike protein is translated in the cell nucleus into messenger RNA. The messenger RNA is released into the cytosol, where it is translated into the spike protein. The spike protein then migrates to the cell surface, where it is recognized by the immune system as a foreign antigen. This elicits the production of antibodies, the activation of CD4 positive helper T cells, and the activation of CD8 positive cytotoxic T cells, killer cells, which is a hallmark of all gene-based vaccines. A potential drawback of adenovirus-based viral vector vaccines is that the risk of pre-existing immunity, such as neutralizing antibodies against the viral vector, which can limit effectiveness. If the immune system clears the vector before it can get into cells, that is a big problem. A pre-existing immunity against the viral vector can be more common in some geographical areas than others, rendering a vectored vaccine more or less effective depending on the region. To avoid the risk of pre-existing immunity, the Astrazeneca vaccines uses an adenovirus that infects chimpanzees instead of humans. However, even with a chimpanzee adenovirus, there might still be cross-reacting pre-existing immunity to human adenoviruses. Immunity against the vector virus can also develop after the first shot in a two-dose regimen. For this reason, the Russian Sputnik V vaccine uses an adenovirus 26 for the first dose and an adenovirus 5 for the second dose. AstraZeneca and the Gamaleya Institute in Moscow plan to run a study exploring a first dose with the AstraZeneca vaccine and a second dose with the adenovirus 26 and vice versa. The Johnson & Johnson vaccine uses an adenovirus 26 strain. In interim analysis of their phase 3 data, respectively published in The Lancet, the efficacy of the AstraZeneca vaccine used in a 2-dose regimen was about 60%, and the efficacy of the 2-dose regimen of the Sputnik 5 vaccine was about 91%. Interestingly, the AstraZeneca vaccine seems to work better than when the dosing interval is longer, between 4 and 12 weeks. Johnson & Johnson recently announced in a press release that according to an interim analysis of their phase 3 study in the US, the efficacy of their adenovirus 26-based vaccine administered as a single dose was estimated at 72%. Overall, the vaccine safety of the viral vector vaccines currently appears comparable to that of the messenger RNA vaccines. So far for gene-based vaccines. Another family of vaccines are the protein-based vaccines. They include whole inactivated killed vaccines, as in the flu shots, subunit vaccines, as in the hepatitis B vaccine, and virus-like particles vaccines, such as the human papillomavirus vaccine. Inactivated or killed coronavirus vaccines are produced by the state-owned Chinese company Sinopharm and by the private Chinese company Sinovac. Inactivation of the coronaviruses is achieved by applying a chemical called beta-propiolactone to the viral surface. The Russians are also developing an inactivated virus vaccine. A protein-based vaccine for which phase 3 data are currently available is the vaccine produced by the US-based company Novavax. The Novavax vaccine uses stable pre-fusion coronavirus spike proteins assembled into nanoparticles, also called virus-like particles. The vaccine is administered in 2 doses and contains an adjuvant for an increased immunostimulatory effect. In a press release, Novavax announced that according to an interim analysis of their UK phase 3 study, the efficacy of their vaccine was estimated at 90%. Now, a big subject currently are the emerging variant strains of SARS coronavirus 2. Such strains are found in Britain, in Brazil, in South Africa, in Japan, and other countries. Recent in vitro studies carried out by Pfizer-BioNTech and Moderna suggest that their messenger RNA vaccines preserve their efficacy against all the coronavirus variants tested. The Moderna National Institutes of Health study reports results from an in vitro neutralization study of sera from individuals vaccinated with the Moderna vaccine in preprint. The results show that the vaccination produced neutralizing titers against all key emerging viruses, including variants B117 and B1351, first identified in the UK and in the Republic of South Africa, respectively. However, the South African variant represents an important challenge, since it carries 10 mutations in the spike protein, with 3 mutations located in the receptor binding domain, among which a key antibody escape mutation in position 484. Thus, a 6-fold reduction in neutralizing titers was observed with the South African variant relative to other variants. The South African variant was, however, fully neutralized, albeit at lower dilutions of sera, 1 in 300 versus 1 in 1800 for other viral strains. Despite this reduction, neutralizing titers with the South African variant remain above levels that are expected to be protective. The existing messenger RNA vaccines are likely to still prevent serious illness, but it may be more difficult to prevent milder disease and transmission. However, the lower neutralizing titers observed in these studies may suggest a potential risk of earlier waning of immunity to the new South African strains. To address this risk, an additional third booster dose may be considered beyond the primary two-dose regimen to further increase neutralizing titers against emerging strains. The third dose may be either identical to the initial vaccine or else customized for the South African variant. Moderna is advancing its vaccine candidate specific to the South African variant into preclinical studies and a phase 1 study in the US. Since the study will be performed in the US, this suggests that they will be looking at in vitro data from neutralization studies. Meanwhile, Pfizer-BioNTech posted on the preprint server bioRxiv the results of an in vitro study testing sera from 20 vaccinated people for neutralization against the British and South African variants. They noted that for 1/3 of the serum samples, neutralization titers were half as high for the South African variant than for wild-type virus. This corresponds, however, only to a small reduction of neutralization titers for the South African variant. So far for in vitro studies. But what about real-world data? In small vaccine trials conducted in South Africa, the US-based companies Novavax with their protein-based vaccine and Johnson & Johnson with their adenovirus 26-based viral vector vaccine have provided real-world data on how their vaccines perform against the new variant there. In a press release, Novavax reported an efficacy of 49% for their vaccine candidate in South Africa, with most vaccine failures attributed to the South African variant, compared to a 90% efficacy in their UK study, so 49% versus 90%. As to Johnson & Johnson, they report in a press release that their adenovirus 26 vaccine candidate, administered as a single dose, was 57% effective in South Africa, compared to an efficacy of 72% in the US. 57% versus 72%. Fortunately, the gene-based vaccines, i.e. RNA, DNA, and viral vector vaccines, are relatively straightforward to redesign for a new variant. Both Pfizer-BioNTech and Moderna have announced that it takes them about 6 weeks to develop a modified vaccine. This, however, does not take into account the amount of time it takes for the conduct of safety trials, such as preclinical trials, phase 1 trials, and the approval of the modified vaccine by health authorities. So I will stop here, as was agreed upon, to give you a chance to ask questions. And these questions will be looked at by our friend and colleague Guillaume Fournier, and he will read them to us. I think that's how it works.